So then we moved to a catch trial where the feedback was randomized, but feedback was still delivered to the animal during the catch trials, but the randomized feedback meant that the feedback was no longer related to the motor cortex ensemble dynamics. And you can see, in this case, they follow each other rather nicely. So this argues that the rat was using the feedback as a signal, meaning that a trial was occurring, but was not actually paying attention to the subtle features of the feedback. And in the other two rats, we noticed something rather alarming. You can see on the catch trials where there was no feedback and unrewarded, the animal is actually doing better on those trials. Why is that? Yeah, it kind of bugged me for a while as well. But if you stimulate 10 milliseconds out of 90 milliseconds, during that stimulation artifact, I am dropping my detectability of spiking activity. So I'm dropping my detection of spiking activity by 11%. That drop in spike detection is also fed into the algorithm. So when there's actually feedback trials, the task is actually harder for the rat because it has to modulate its spikes 11% more to generate a detectable signal via Greg's algorithm. If we look specifically, you can see that there's a nice transient increase in the no feedback trials that is much larger than the feedback trials. And if we remove the feedback altogether, you see this, the modulation is much, much stronger on some representative histograms. But then I thought, okay, the rat's just paying attention to the initial go cue and not really paying attention to the feedback. So I dropped the initial go cue. And so all the feedback delivered during the response was the only indication to the animal that a trial was occurring. And in that case, you can see that modulation is weaker, but nonetheless, the animal could do the task. So even though the task was harder with the feedback, the animal could still do it. And the other rat of the three rats had essentially the same results. Let's talk about some exciting stuff. I told you I'd play Danzig at my defense. Yeah. Okay, can anybody tell me what's going on in the bottom? Why is the rat hitting the lever? There's no food. Doesn't seem to have any relevance to the animal. Anybody know? Anybody know their neuroscience history? Any grad students in this room changed the neuroscience with their first experiment in grad school? James Olds did so, discovering the MFB, which is the part of the brain, it's part of the reward pathway. You stick an electrode in this brain area, and rats will actually self-stimulate because it's rewarding. The way it was actually discovered was an accident. James Olds was trying to, at the time they had discovered the role of the brainstem in attention and alertness. So James Olds was trying to stimulate the brainstem, the reticular activating system, during a behavioral task to see if it made the rat learn faster. He was a grad student, it was his first surgery, he messed up the implant, but then noticed that the rat kept returning to where the stimulation was. And so he had the foresight to investigate this further, and then he systematically mapped out. So I didn't revolutionize neuroscience with my first bad experiment, but maybe some of you guys can. And actually the histology for that first rat is not available because he wasn't good at histology and ruined his slices. Okay, so what I showed here is motor cortex ensemble modulation, auditory cortex microstimulation as the stimulus, and MFB as the reward. So maybe we could combine all these, where the whole task is done solely through brain electrodes, where the stimulus, the action, and the consequence, to use Skinnerian terminology, is solely through brain-machine interfaces. Okay, how this works? I record motor ensemble baseline activity, then deliver current into the visual cortex, monitor motor ensemble activity. The animal has to increase its motor cortex ensemble discharge rate to be rewarded, then stimulate the MFB if the rat indeed does so. In the lab, we call this the matrix rat, or the neo rat, because the whole task is through the brain electrodes. Kind of hard to get all three of these electrodes working, but in one rat I was able to get it. The matrix was online for one day. You can see I tested whether the MFB was self-stimulating prior to the behavioral session by rewarding a rat with MFB stimulation every time it's put its nose in a hole. You can see in 3 minutes he put it in about 20 times. If you just leave a rat in a cage, he'll probably put his nose in once or twice if it's not related to any reward. Here's the histology of the MFB. You can see with the visual cortex intracortical microstimulation, there's a nice increase in firing rate. You could say, Tim, the MFB is close to the internal capsule. Maybe you're antidromically stimulating the motor cortex via your MFB stimulation. Well, I would say, well, let's do the PSDH, and you can see that the motor ensemble activity is before the MFB stimulation. So it argues that the animal was doing the task. Okay, so that's it. Sweet, not too much. So in my first experiment, I showed that the cingulate cortex is a compelling alternative site for a potential neuroprosthetic device. In my second experiment, I showed that if you wanted to combine local field potentials and spikes, you would band pass the local field potentials above 10 hertz. And I showed that in the third experiments, rats can detect intracortical microstimulation, appropriately modulate the motor cortex, but I really feel that the hardware needs to be improved before it actually becomes a reality. So, some of the more pessimistic people in the audience would say, Tim, oh Tim, you just played games with rats for 6 years. If we're really going to make neuroprosthetics a reality, there's some more fundamental electrode issues we need to address. And I would agree with you. Would totally agree with you. After 6 years, I realized that for neuroprosthetics to become a reality, there's a glaring problem that needs to be solved, the electrode problem. You stick electrodes in a rat or monkey or a human, they fail within 3 to 6 months. No one solved it, though we are working on it in our lab. And until that is solved, we are simply just playing games and doing exciting demos in animal models. So I really feel that if you're a young grad student and you're interested in neural engineering and neuroprosthetics, don't train animals. Don't try to figure out what the animal's perceiving. Try to just do the boring and mundane experiments of just coding electrodes and seeing if it improves electrode viability. I'm not making fun of you, Kip. I think it's an important experiment, but you know it's boring. Those are boring experiments, you know? Stimulation hardware. I don't even know what the artifact is. What is it? Why, if I put electrodes in the brain and stimulate, do I get a recording artifact on my recording electrodes? Is it because electricity is being delivered? I mean, there's electricity being delivered across my jaw muscles. How come I don't get an epileptic seizure every time I chew gum? I don't know that. Also, if some of you saw Ed Boyden's talk, perhaps light-based stimulation could have completely avoided the intracortical microstimulation artifact. Because, as John knows, and any of the rest of you in the room that study sensory neuroscience, the brain does not encode features by firing for 10 milliseconds out of every 90 milliseconds, right? Everybody knows that, right? Your brain is not quiet every 80 milliseconds. But I had to use that interleaved feedback scheme in order to simultaneously record and stimulate. So you could even argue, what the hell was the animal even perceiving to begin with? And so that problem needs to be solved as well.